Hello, my name is Leah Campbell. I am a certified genetic counselor and medical science liaison with Baylor Genetics. Today, I will be discussing our global metabolomics assisted pathway screening, or global MAPS. I want to give a preview of what we'll cover with this presentation. We'll begin by exploring the background of inborn errors of metabolism and why comprehensive metabolic screening is so important. From there, I'll walk through the advanced technology that powers global maps. After that, I'll explain our specimen requirements. And then finally, we'll discuss our reporting framework where we convert raw data into clinically relevant findings using Z-scores and a structured categorization system. By the end of this presentation, you'll understand how global maps complements existing diagnostic tools and enhances patient care. To begin discussing global maps, it's important to first understand the underlying conditions it is designed to help detect, that being inborn errors of metabolism, or IEMs. Inborn errors of metabolism are a large group of genetic disorders characterized by disruptions in normal metabolic pathways. These disruptions are most often caused by a defect in a single gene that encodes an enzyme. This enzyme typically functions as a biological middleman, converting one substance into another in the body. When that enzyme is absent or dysfunctional, that conversion process breaks down. As a result, three main problems can occur. A toxic substance may accumulate. A normal substance may build to harmful levels that interfere with cellular function. Or the body may lose the ability to synthesize essential compounds needed for growth, energy, or cellular maintenance. These disorders may also be referred to by other names, including congenital metabolic diseases and inherited metabolic disorders. Regardless of the terminology, they all refer to the same fundamental issue, that being a breakdown in the body's chemical processes at a cellular level. While each inborn error of metabolism is rare, collectively they are relatively common, with an estimated combined incidence of 1 in 800 to 2,500 live births. Because metabolism is a basic cellular function, these disorders often affect multiple organ systems, especially vital ones like the brain, liver, kidneys, and muscles. Given the clinical variability that we can see, and the potential for subtle or delayed presentations, many inborn errors of metabolism go undiagnosed for years. This is why advanced screening and diagnostic tools like global maps are so essential. They allow us to identify biochemical imbalances early, even in patients with complex or unexplained symptoms, potentially leading to earlier intervention and improved outcomes. To appreciate the value of global maps, it helps to first understand the concept of the metabolome. You may already be familiar with other omes: the genome, which encodes our DNA sequence, proteome, the full complement of proteins, and the transcriptome, or the full set of RNA transcripts. The metabolome is the collection of small molecules or metabolites that are present in a biological sample. Unlike the genome, which is essentially fixed at birth, the metabolome is dynamic. It sits at the end of the central dogma as the chemical products that proteins act upon or create. Because of this, the metabolome reflects real-time biochemical activity in the body, and it can shift dramatically in response to cellular function, environmental factors, diet, stress, or medications. For example, a patient during a seizure episode will display a markedly different metabolic signature than when neurologically stable. Similarly, changes in diet or the introduction of a new medication can produce rapid and measurable shifts in metabolite levels. This sensitivity to physiological and pathological change makes the metabolome an exceptionally powerful diagnostic window, one that complements genetic testing by revealing what is actually happening biochemically in the moment. By capturing this snapshot of metabolic activity, Global Maps leverages the dynamic nature of the metabolome to detect pathway disruptions, identifying toxic accumulations or deficiencies, and to guide clinical decision making. Global Maps is a large scale, semi quantitative metabolomic screening platform that's been developed in collaboration with Metabolon. In each patient sample, we identify and semi quantify over 700 known metabolites. These metabolites range in molecular weight from approximately 75 to 1,000 Daltons using advanced methods. Rather than presenting isolated analyte values, Global Maps employs a pathway based approach. Each individual profile is compared against a healthy control reference cohort of 200 to 400 samples to detect outliers and patterns of biochemical dysregulation. Metabolite levels are expressed as z-scores, normalized to the control population, so clinicians can readily see which compounds deviate most significantly from the expected range. Global Maps also supports supervised differential analysis across multiple groups, enabling the discovery of novel biomarkers and insights into disease mechanisms. The Global Maps platform detects a comprehensive spectrum of metabolite classes. It quantifies amino acids ranging from well-characterized analytes such as phenylalanine and leucine to their diagnostically significant downstream metabolites. In addition, it surveys carbohydrate intermediates, a diverse array of lipid species, nucleotides, essential vitamins, TCA cycle metabolites, and xenobiotic compounds, including pharmaceutical agents and diets. Our untargeted metabolomics workflow begins with metabolite extraction, in which small molecules are enriched from biological samples using a methanol solution. This step precipitates proteins and isolates the low molecular weight fraction for downstream analysis. Extracted samples are then analyzed on 4 separate liquid chromatography mass spectrometry runs using a system equipped with hydrophilic interaction columns and a high-resolution mass spectrometer. 2 runs employ positive mode electrospray ionization, and 2 use negative mode electrospray ionization. This dual mode approach maximizes the coverage of diverse chemistries across the metabolome. During each run, metabolites are separated by retention time and then ionized for mass analysis. The high-resolution mass spectrometry measures both accurate mass and tandem MS fragmentation patterns, which are matched against a curated library of over 2,500 human small molecule metabolites and more than 4,000 total compounds. This includes xenobiotics and drug metabolites. Every library entry is characterized by reference standards with known retention times, nominal masses, and spectral fingerprints, ensuring confident metabolite identification. Quantification is semi-quantitative. Peak intensities from patient samples are normalized to a control reference population and then converted into z-scores, which are standard deviations from the mean. These z-scores are interpreted both on a per metabolite basis and in the context of the global biochemical pathways, enabling detection of perturbed metabolic networks. As part of this process, z-scores may be interpreted within the context of a Cytoscape plugin that allows data comparison and analysis of individual samples and groups of samples to provide a more global assessment of the metabolic disturbances in the patient or the patient population. Every filled circle on this map represents a molecule identified in this analysis. As you can see, this pathway analysis allows us to explore multiple biochemical pathways in a single analysis, allowing a comprehensive coverage of the biochemical pathways involved in small molecule metabolism. Global Maps serves as a cost-effective supplement to both newborn screening and targeted Metabolic tests, offering a general screening tool for patients with highly heterogeneous, undifferentiated phenotypes. It can be particularly valuable for individuals presenting with intellectual disability, developmental delay, hypotonia, seizures, or a diagnosis of autism spectrum disorder, groups in which traditional testing and screening may not always result in a diagnosis. Moreover, global maps can detect mild or late onset metabolic disorders that often evade routine laboratory assays or newborn screens and may remain undiagnosed for years, sometimes requiring multiple rounds of testing. By casting a wide net over hundreds of metabolites, this approach not only broadens identification across the phenotypic spectrum, but also facilitates earlier intervention. In a recent study by Smirnov et al., a systematic analysis of molecular genomic and metabolomics data demonstrated a diagnostic yield of 12%, underscoring its potency in diagnostic strategy in complex clinical cases. Despite its broad coverage, Global Maps is subject to limitations that every clinician should bear in mind. First, the assay is optimized for small molecules between 75 and 1,000 Daltons, and therefore cannot reliably detect analytes outside this range, whether smaller than 75 Daltons or larger than 1,000. As a result, proteins and large peptides, complex oligosaccharides, large lipids, and elemental ions such as potassium or sodium fall outside the scope of this test. Likewise, it's not intended to screen for conditions driven by high molecular weight substances, including lysosomal storage disorders, congenital disorders of glycosylation, mucopolysaccharidoses, or mucolipidoses. Certain metabolites also require specialized extraction or chromatographic methods. Homocysteine, for example, must undergo reductant treatment to be measured accurately, so it's not captured in the standard workflow. Moreover, Global Maps delivers semi-quantitative results expressed as z-scores relative to a reference population rather than absolute concentrations, and it's not suitable for acute metabolic crisis assessment. Pre-analytical variables can further influence results. Patients receiving TPN, adhering to special diets, taking nutritional supplements, or on certain drug therapies may yield confounded profiles. Nevertheless, by delivering a comprehensive snapshot of hundreds of small molecule metabolites, Global Maps remains a valuable complement to targeted biochemical assays and genetic testing, providing critical insights into metabolic pathways that otherwise may go unrecognized. For specimen collection, we accept two sample types. Our preferred specimen is plasma, submitted under test code 4900, which provides the most consistent and high-fidelity measurements across our metabolite panel. In certain clinical scenarios, such as when evaluating for lysinureic protein intolerance, we offer urine testing under test code 4901, as it can yield diagnostic insights that plasma alone may not capture. Once the sample arrives in our laboratory, you can expect a turnaround time of approximately 21 days. In our reporting framework, we emphasize clinical relevance rather than raw concentration values. Each metabolite and biomarker is evaluated against a reference population and classified into one of four categories. First, we identify significantly altered analytes, those with z-scores greater than +2 or less than -2, that may be directly related to the patient's phenotype. Next, we flag rare analytes, compounds observed in fewer than 5% of all prior specimens, whose unexpected presence might point to a unique metabolic disturbance. We also report absent common analytes, molecules that are normally detected in over 99% of samples, missing in that specific patient's profile, since their absence can suggest deficiencies or disrupted pathways. Finally, we highlight nutrition or treatment-related analytes, where significant deviations are likely attributed to diet, supplements, or medications rather than primary disease processes. Here you see how the report is structured, concise but informative. These reports are designed with both generalists and specialists in mind, making them accessible and actionable for all. Rather than overwhelming the reader with raw data, it groups findings by potential relevance to the patient's phenotype, allowing for a focused follow-up discussion. Thank you for your time and attention. We believe Global Maps is a critical addition to the metabolic diagnostic toolkit, particularly for patients with nonspecific or complex phenotypes. Please let us know if you have any questions. We're happy to follow up after watching this recording.